And it seems that we're recording now. So like I said, this is week 8 evening session that we're having a look of. And I apologize, we're starting a little late. I already mentioned, but just for the continuity or for the sake of being a little bit more consistent, we're starting a little late, and because we have some technical issues, I have uploaded you a study guide that probably contains a little bit more answers than what I'm gonna post in the future or normally in the next day or so. I'm gonna adapt that study guide a little bit. What I asked was that if you could log in to or you're probably already logged into your Blackboard, and then go to our course modules, and on module for week 8, you should be able to find on the lecture study resources a study guide. And we're going to use that study guide today for our review session. And like before, all of the questions that are going to come up in the exam are going to be from that study guide. So don't feel that there's anything too daunting coming up. If you have a chance to either download the study guide, that's great. If you're able to print it for yourself, that's great. I know that we're running on a short notice, so if you need a moment, I will take it a little slower to start with, but I would ask you to have a look if you're able to access that. And I'll jump back to our PowerPoint presentation. Let me get there. And we should be back to the PowerPoint. So what I was hoping to do was really kind of have a quick look where we're at on our course, have a look what's this week, what are we going to do? We're going to talk a little bit about the exam 2, lab practical 2, and review the material by using that study guide for the exam. For the lab practical, it's going to be following the same format as before. There's going to be blanks that you'll be filling, and it's only blanks, so there's nothing out of ordinary. I know that last time initially the software gives you some grade, which is often mistakenly harsh, because I have to manually go and review those. So if you give me a little bit of time after the exam, the lab grades get finalized. Last time I went, I think, 3 times through them for each one, just so that it was consistent. So no one got graded harsher or easier than others. So that's the plan for today. And while I'm getting started, if you can have a look, if you're able to get a copy of the study guide for the lecture material from week 8 resources. Just to have a quick look, I've got a wrong week 8. And we're luckily half done almost. So where we are now, we're approximately halfway through the course. We have talked, I think, what I would describe as the hardest material on this course. We have looked at the blood, we have looked at endocrine system, we have looked at heart, and we have looked at blood vessels. And the last chapter, lymphatic and immune system, was probably quite a lot to take in. And I realized that, especially since that's kind of a topic where I think that to really understand, we need to dive in quite deep. So as you'll notice as we review through the material, I'm not expecting you to be experts of that field. But this week we're looking at the exam 2 and lab practical 2. So those are the only assignments or only pieces of activities that I'm asking you to do. There's no traditional chapter material or no traditional assignment. So just the exam and the lab practical. So I want you to feel that you have enough time to prepare for that and take it without having to rush. You'll notice that the exam isn't up there yet. I expect it to be up early tomorrow morning, but I'll give an extra day of submission time for that in regards to that. You'll see a notice or an announcement of that going out. But I've said before, and the same goes now as well, if you do have some assignments from previous weeks that you would like to give a go, you didn't get a chance to do that before a deadline, or you didn't grade as to the standard that you're wanting to reach a grade, I'm open to reopening those. So contact me. I know that there's few messages. I'll hopefully get to them early tomorrow morning on our Canvas messaging system. So that's the easiest. If you send me a message with the details of the assignment, I'm very happy to give you a chance to get those points as well. Really, on these online courses, the big deal to get a good grade is to just systematically go through all of the elements that earn you points. It's up to you to kind of assess what is most important, what is not. But assignments add up to a nice amount of points, and most of the time with assignments, you can kind of really spend your time and get those points. I have also here typo. So we're looking at the, of course, exam 2 and lab practical 2. So both of these I initially put at the end of the week, but of course, since the exam 2 is gonna be opened late, I'll extend them by a day for both. And you'll see the points that they carry. So really, lab practical is worth of 5 weeks worth of quizzes. So that gives you an idea, an exam worth of 10 weeks worth of quizzes. So they do carry a little bit more points, but like I said, there's nothing new. There's nothing that you wouldn't have seen before. So I don't want you to worry about that. But they're definitely something that you want to get done. You don't want to just give a miss to one of those. There's going to be a study guide. This picture is from the previous one, but there's a study guide that you should be able to find on the Canvas module for week 8. So have a look there. If you're able to download or print that for yourself, we're going to use that. And I've loaded a little bit of different. I've loaded it by chapter, lab practical study guide as well. It's Word documents, but I'll have a look if I combine them or what I'll do over tonight. So the layout of those might change a little bit, but the material is there, what to pay attention to. So similarly to what we had on our lab 1, you also have a practice for this lab 2 available on your Canvas course. I am going to be uploading again, I think overnight, handouts for all the lectures, the slides that are not exactly the same, but very similar to what I've had on our lecture videos. The slides are exactly from your textbook. So I think that at that time, this time we're ready to look at the study guide. So let's jump there. If you have it in front of you and you're able to fill in answers there, that might come in handy. And let me just open up the right version. So what we'll do, we'll discuss through it. I'll show some proposed answers, but again, of course, there's many ways to answer to those. So I am not saying that what you're seeing is the only correct answer. At any time, if you have questions, just shout out or post them up. But I think the biggest one that you're probably looking for is to get a little bit of those answers. So let's go through. And what you'll notice that for the last chapter 20, I think. These chapter numbers change a little bit from textbook to another, or a version of the textbook to another. But the last chapter where we talked about lymphatic and immune system, for those questions, I have all the answers, or for all that part of the study guide, I have all the answers for you on Monday evening session already in that study guide. And what you'll notice that probably either tonight or early tomorrow morning, it's going to update into a version where there's no answers for the last chapter as well. But because we're started a little bit later than what I had intended, I wanted to load a version where there's answers for that, so we can focus on the probably the hardest material what's coming up. So we started with the chapter about the heart, and what we'll notice that the term that we often use when we talk about heart sciences, or we talk about heart surgery or heart research or heart medicine, is the term cardiac. If someone doesn't have an access to that study guide, I'm just quickly going to say, shout out now. I can even send it to you directly here on this web meeting format. But if I don't hear anything, I assume that you've been able to access the study guide. So the other thing that we noticed when we started talking about the heart was that there were two circuits that we were looking. And those two circuits that we saw were pulmonary and systemic circuits. We, of course, remember that the pulmonary circuit is all about the lungs. And the function of this is to take de-oxygenated blood, so blood that's low in oxygen or almost short of oxygen, and take it then to the lungs, get it oxygenated there, and then bring it back to the heart. And the flow of the events or the steps here is that we start with the right ventricle, where the deoxygenated blood gets pumped to the pulmonary trunk. Pulmonary trunk gets divided into left and right pulmonary arteries. They divide further to arterioles and eventually to capillaries at the lungs. During there, or once we get there, the exchange of gases take place. So we are picking up oxygen and getting rid of carbon dioxide. And we'll talk about that process in greater detail in our chapter where we look at the lungs. And the blood that returns from the lungs to the heart follows the path of leaving the lung capillaries to the venules and then pulmonary veins and returning to our left atria. This blood is now going to be oxygenated since we have done the exchange of the gases there. The other circuit that we'll see was the systemic circuit. Systemic circuit serves all other parts of the body. So while the function of the pulmonary circuit was to get the oxygen to the blood again, now this oxygen-rich blood gets pumped everywhere in the body, and the path of that is going to be from the left ventricle to the first of our arteries. And first artery that we find is our aorta. After aorta, we move on to the further arteries, eventually to arterioles, and finally to the systemic capillaries. This blood is going to be oxygenated, and it's going to then travel. Once the oxygenated blood has reached the capillaries, the tissues take up the oxygen, and it's going to travel back to the heart as deoxygenated, following the path of systemic capillaries, arterioles, arteries, up the superior or inferior vena cava to the right atrium, back to the heart. So those were the two circulations. I think that on one of our lecture videos, I said that, or weekly live meeting videos, I said that it's almost like two hearts beating as one if you want to be very poetic about that. So we've got the 4 chambers which serve 2 different circuits, and the pulmonary was all about lungs. The pressure on this pulmonary circuit is going to be lower because we've got thin capillaries there at the lungs, so we want to protect them. Whereas the systemic circuit, we have higher pressure on that, and we'll talk about that a little bit later. What I want to show next was that we should start always when we talk about a structure by kind of defining a little bit of its location. Where is it present? So we end up finding that the heart is located within the thoracic cavity. There's an area called mediastinum within there, contains the central parts. Heart also, if you think about it, located between the lungs, and we'll see while we're going to study later on in the next unit the lungs, you'll notice that the location of the heart actually leaves imprints to the lungs as well. The heart is located above, so superior, if we use proper anatomical terminology, superior to the diaphragm. So diaphragm is simply a large sheet of muscle. And we'll have a look when we talk about the respiratory system, more about diaphragm and its function and so on. Often you hear that the way to a man's heart is through their stomach. I would say whether it's a man or a female, it's probably through the intercostal spaces 2 and 5, between 2 and 5 intercostal spaces. So between the ribs 2 and 5 is where the heart is located. And if you want, you can actually identify that by following your own ribs and from there working out where your heart is going to be. So ribs are fairly easy to feel in terms of our external anatomy. So you can identify that what sort of a height at the superior and at the inferior border of the heart, where is it located? The other kind of a rule of thumb is that heart is going to be roughly the size of your fist. So that might be helpful. But we'll notice that heart is located much more into the midline of the thoracic cavity than most people think. So we're almost at the midline and we move just slightly left from the midline. Remember that when we talk about right and left, we always talk in relation to the patient. So patient's left. And what we'll notice that if we had the heart that was almost like a fist, the very pinky finger would be the very bottom of the heart. And very bottom of the heart contains an area that we call apex. That's the tip of the heart. You'll notice that in some other structures, the tip might be superior part. It's just the sharp end part of that structure. But in heart, it's the bottom tip of the heart. And that's going to be pointing, if you want to think about how it's pointing, it's going to be pointing slightly downward, so slightly inferiorly and slightly left. So if you place the heart, your fist where it would be, it's going to be just off from the midline, inferiorly and left with the head, tip of the heart pointing there. In terms of the layers, we had a look of the coverings and layers that heart had. And let's go through first what they are and then have a look of why do we have them. So let's look at the heart wall first. So heart is made of a muscle wall. And this muscle, if we move from inside the heart going towards the outside the heart, the layer that's closest to the inside the heart and dealing there with the blood that we have flowing inside the heart, that's going to be our endocardium. There's many ways that you can think of. You can come up with your own mnemonics, but endocardium is the innermost layer of the heart wall. Thickest part of the heart wall is going to be myocardium. This is really the meaty layer. Often when I do a lot of research on the heart and heart muscle, it's really the myocardium where we focus. That's the really obvious part of the muscular layer of the heart wall. And the outermost layer of the heart muscle or heart wall is our epicardium. So the order is going to be from endo to myo and then to epi. I think one way, if you're big in geography and you're interested of that epicentrum of an earthquake is always the layer on top of the earth that we see on the surface of the planet, even though normally the earthquakes happens somewhat inside our planet. So epicardium is just the top layer there, or top point there that we see on the surface of there. So if that helps you to remember, that might. If that doesn't help you, don't get confused. We're not expecting you to be expert of geography. Let's have a look of also the sac that holds the heart inside it. There we've got two layers only. We've got innermost visceral layer, and then we've got outermost parietal layer. Between those two layers, we've got pericardial cavity, which is filled with fluid, our pericardial fluid. The function of that fluid is to lubricate these two layers as they slide past each other. One way to think about it is almost to think about Ziploc bag, where you have a little liquid. As soon as you add that liquid into the Ziploc bag, those two layers that form the two sides of the Ziploc bag, they flow or slide past each other much more easily than they would slide if they are dry. So that's the function of the pericardial fluid. So as we see there must be a reason why the heart is within this sac. Well, it did allow the sac and the structure of the sac, for the structures of the sac to slide past each other. But if you look, there's a bigger purpose why we enclose the heart within the sac. Having it in the sac allows us space and ability for that heart to move and change a little bit of its shape without being held back by the structures that surround it, mainly the lungs. So the lungs don't prevent the heart from its movements, and that's thanks to our pericardial sac around the heart. In addition, the pericardial sac, and you'll notice this in a lot of the anatomy, and I think that this is something that I typically at least discuss if you take BIO201. I don't know whether that was addressed in your course, but body is really divided into compartments. And you'll notice that these different body regions are separated. And often the reason for this separation is also to prevent any inflammation from spreading from one part of the body compartments to another. So by enclosing heart into the sac, we keep any inflammation that might be outside the heart from reaching to the heart. Or alternatively, if a heart gets inflamed, that inflammation doesn't spread to the other parts. If you have taken already your medical terminology class, you'll notice that whenever we have a term itis, that's some sort of an inflammation. Well, in this case, it would be pericarditis, when the pericardial sac would be inflamed. What we'll end up noticing on that has a very distinct sound when you listen the heart with a stethoscope. We'll notice that there's a little bit of an issue there from that sound. What happens is that also this pericardial space gets filled with fluid, and the heart's pumping ability is effective. So we don't want to have pericarditis. We want to diagnose it and we want to treat it as necessary. The next page lists quite a few different characteristics that could apply to one or multiple of the muscle tissue types that we've learned. We learned at the very introductory biology classes that we've got skeletal muscle, we've got smooth muscle, and then we had cardiac muscle. And of course, in case of the heart, we're now going to be looking at the cardiac muscle. And if you think of the cardiac muscle cell, some of the characteristics that we saw with that was that the cardiac muscle cell had multiple branches, and these branches were connected with something known as intercalated discs. These intercalated discs allow for substances from one heart cell to pass to another heart cell easily. That can be chemical substances, that can be electrical impulses, that can be various different things. The function why we have these many branches and these intercalated discs are that we want to be able to transmit these signals from one heart muscle cell to another, so we can coordinate them so that they function in uniform, so that each heart muscle cell on that area contracts at the same time. We also noticed that the cardiac muscle cell is only found within the heart. It is uninucleated, so there's only one nucleus in single heart muscle cell, and we end up noticing that these have striations, so those lines between or lines within the heart muscle cell. And we talked about the cause of those lines when you looked at the chapter on the muscles, I believe, on BIO201. Next thing that I want to talk with you kind of summarizes the really big take-home messages of the heart anatomy. So let's have a look. First thing that I did, I listed the flow of the blood within the heart. And to really understand the flow of the blood, we need to learn what these structures are that I'm referring to. So we're going to first learn those structures, and then this flow of the heart will make sense. But we'll notice, again, we're seeing this pulmonary circuit, which was taking the blood to the lungs. And then we'll notice this systemic circuit, which is all about taking to the rest of the body. And we're focusing now only on what happens within the heart. We're not looking rest of that circuit at this point. You remember during our lecture videos, I said that this is, if you draw a box that's divided into 4 smaller parts, that's enough for showing what a heart is. Heart is nothing but 4 different spaces. So 4 different cavities that we end up finding there. And this diagram, again, remember, we're looking from the point of view of the patient. So at the very superior part of the heart, we have our atriums. So we have right atrium and left atrium. At the bottom, so inferior part of the heart, we have our ventricles, right ventricle and left ventricle. And ventricles are separated by interventricular septum. And this term will become important for us later on as we're doing our review. But at this point, I would like you to know that we have right atrium, right ventricle, left atrium, left ventricle. And what we'll notice that the flow of blood is that the blood returns from the body as de-oxygenated to our right atrium. This could be superior or inferior vena cava that carried the blood there. And from the right atrium, blood flows to right ventricle. And this blood has to go through right AV valve. The other name that we know there is often tri-cuspid valve. That describes the anatomy of these valves. There's 3 different cusp-like structures, kind of a leaflet-like structures. So 3 of them, so that's why tri. And once blood has passed this valve, it cannot flow back. So that's the function of the valves. Well, now we have the blood at the right ventricle. Right ventricle contracts and pumps the blood through pulmonary valve, and from there to the pulmonary trunk. And now blood goes to the lungs, and there it gets oxygenated again, and it returns from the lungs to our left atrium. Left atria, there's actually going to be 4 openings within left atria where the blood returns from the lungs, 2 from the left side, 2 from the right side. And from left atria to left ventricle, we have to pass through another valve. And that can be either our left AV valve, or our mitral valve, or our bicuspid valve. These all mean the same thing. The bicuspid again tells us about the anatomy of this valve. So 2 leaflet, 2 cusps on this valve. Mitral is another term that's commonly used. And I would not say that the LAV or right AV would be a bad term either to use. Remember that AV stood for atrioventricular valve. Well, once the blood has gotten to the left ventricle, thing that we'll notice that the left ventricle will have the thickest muscular wall of all of these chambers that we're seeing. Well, from left ventricle, blood heads to the systemic circuit. Remember, this is now highly oxygenated blood. And to get to the first part of the systemic circuit, which is going to be our first artery, that's going to be our aorta. The blood has to pass through aortic valve. So there we have our chambers of the heart and our valves of the heart. This is the most simple way of looking at the heart. If we want to look at it in a little bit more detail, a diagram that looks something like this might be familiar when you've been looking at the heart in the past. So what we'll notice, I've just zoomed out quickly so I can show what you'll notice that we have again these 4 chambers. Now we have added a little bit more anatomical detail there, but I want to highlight a couple of structures. We've got our apex, so the very tip of the heart here, pointing downwards, so inferiorly, and pointing to the left. So that's the tip of the heart that I was talking. And you'll also notice that the ventricles are separated by interventricular septum. These terms are already filled in for your review sheet. What else do we see? We see superior and inferior vena cava bringing blood that's going to be de-oxygenated to the right atrium. And let's start there. I'm going to zoom just a little closer in case anyone's having a hard time seeing it on the screen. So we're going to start the study of the flow of blood in the heart, compare it to that box view that we saw earlier on the 4 boxes. This is the same. I've just added a little bit more detail here. Right atria, where the blood comes from the systemic circulation, this blood is going to be de-oxygenated, so little oxygen in this blood. It receives the blood from the inferior vena cava and superior vena. Well, superior and inferior vena cava fill the right atrium with blood, and once right atrium has been filled with blood, then the blood moves from the right atrium to the right ventricle, and this is complemented by the contraction of the right atria as well. For that passage, we had to go through the right AV valve. It was also known as tri-cuspid valve. So this valve here. And remember, this valve allows only one directional blood flow from here. What else should we know? Well, once the deoxygenated blood has pooled to the right ventricle, it heads to the, and I have the labels here wrong way around for the aorta and pulmonary trunk, so you might want to make a note of that. I'm going to change them to the review sheets, but notice that these are wrong way around. So aorta should be where the pulmonary trunk is, and pulmonary trunk should be where the aorta is. So the blood flows from the right ventricle to the pulmonary trunk through pulmonary valve. So just make sure that you fix that. I will fix it on the updated version of the review sheet, but if you already downloaded or printed a review sheet, so just change the aorta and pulmonary trunk the other way. So now the deoxygenated blood is going to go up the pulmonary trunk to the lungs. It's going to get oxygenated there, and it's going to return back to the heart through our pulmonary veins. So this blood will be now oxygenated. This is the only time in the veins that we see oxygenated blood flowing in the veins. Normally it's going to be de-oxygenated, but pulmonary circuit makes an exception of there. And there's going to be 4 openings, 4 pulmonary veins that open to left atrium. And the blood flows from left atrium to left ventricle, again through a valve, so left AV valve, also known as mitral valve, also known as bicuspid. We saw that earlier. And if you look at this diagram, the left ventricle really has the thickest muscle wall around it. That's because left ventricle squeezes the blood out stronger than any other chamber of the heart. And the blood will flow up to the aorta through aortic valve that we'll see here. So again, make sure that you switch these around, the aorta and pulmonary trunk. So that's the same as what we saw here, just shown with a little bit more anatomically accurate detail. So all you need to fill in here for your review notes is the right atrium ventricle and left atrium left ventricle, and then the valves. So we start with right AV valve, also known as tri-cuspid valve, our pulmonary valve, our left AV valve, also known as mitral valve, also known as bicuspid valve, and finally our aortic valve. So that's our heart anatomy. I've added a little bit of extra notes here. We remember valves, their function was to facilitate one-way flow of the blood, so unidirectional flow of the blood. And what really causes a valve to open or close is the pressure changes. It's the pressure that changes that causes the heart to or valve to either open or close. Sometimes we end up having issues with the valves. We can have a completely failed valve, in which case that valve doesn't work at all. It's just open all the time. It doesn't close properly. In that case, there's going to be backflow of the blood to the direction where we don't want it to go. As a result of that, the heart ends up pumping that same blood through that valve back and forth. So the heart and its function of pumping blood onwards to one direction of only is not going to work. And probably depending on the severity of that, individual might be fine, but heavy exercise, other issues would be significant risks. So we want to pick that up. If the heart is narrowed, the valve doesn't, or if the valve is narrowed, the valve doesn't open properly. So now, to get all that blood through that valve, heart has to contract even more stronger, even stronger than before, and this insufficient opening puts stress on the heart. So both of these we'd be able to hear again with a stethoscope when studying the heart and heart sounds. Couple of other names and structures that I want you to be familiar with. There's auricles. These are the projections from atria. They're kind of empty sacs at the top of the atria. You can consider that their function is that they only expand when the atria is full of blood. So they give us a little bit of extra space for the atria by being there. And then when atria is full, they get flat again, they collapse. Another structure that's worth noticing inside our ventricles is taberculae carnae. These are projections from the wall of the ventricles, and these projections serve the purpose that blood doesn't get to pool and stay still within these ventricles. So when blood stays still and doesn't move, it tends to start clotting. So we don't want blood clot forming within the heart by any means. So that's why we have these projections from the ventricular wall. They help kind of the blood to get mixed. We talked about the septum, interventricular septum. So septum referred to a wall of some kind. In this case, it was to separate the right and left side of the heart. So interventricular septum separates the ventricles of the right and left side of the heart. We can sometimes, I mentioned earlier on that we can have issues with heart valves. They can be replaced if we cannot fix them through a surgical intervention. We can put a fully mechanical heart valve. That's one option. But fully mechanical heart valves are not ideal because if we're talking of a younger patient especially, because heart keeps growing throughout your age. We use mechanical heart valves preferably only for older individuals, or if we use mechanical heart valve for a younger individual, we know that we have to do another surgery to change the heart valve most of the time. So that's why a heart valve that comes from a living organism is usually better. We can take a heart valve from another individual who has died, so from a cadaver. This would be known as allograft. Or we can take a heart valve from another animal than human. Often pigs are what we use, so pig heart valves are what have been traditionally transplanted to humans, and these are known as xenografts. Where would you have heard about allografts and xenografts? This is at our last chapter when we talked about the organ donation. So I'm kind of combining terms from two chapters here. There's a few other structures. We really didn't talk about the embryonic development of the heart, so I wouldn't be too bothered about this, but I want you to be familiar that you've seen these before. So fossa ovalis is something that separates the right and left atria from each other. And when you have an embryo before you are born, or before individual is born and is still developing, we don't really have functional lungs. So it doesn't make sense to pump blood to the lungs when lungs don't really work. So we bypass that entire part of doing the pulmonary circulation by pumping blood directly from right atria to left atria. However, when you are born, that little hole that facilitates this flow from right to left atria, that closes at the moment when you're born. And that's because of the anatomy, how this hole is built. We can have sometimes individuals where this closure is insufficient or doesn't happen at all. And these individuals definitely require medical intervention. But most of us are fossa ovalis fuses. We might be able to see some remains of former fossa ovalis on an individual's heart, but it's not an opening there that we could anymore use. You can also see within the right atria coronary sinus. This is where the blood returns from the coronary circulation. So we'll look at coronary circulation a little bit later. So it's where the heart itself gets blood. So that heart blood needs to be returned to the heart and circulation somehow. So that's through coronary sinus, which is found in the right atria. And of course, it's found in all ages of an individual. Ligamentum arteriosum is another embryonic structure if the blood has flown from the right atria to right ventricle and then to the pulmonary trunk, but we have not yet been born. We can still pump the blood back to the heart without going to the pulmonary loop by this structure called ligamentum arteriosum. That has usually some sort of a remain in an adult, even that we can see if you do a good dissection work. So that's little structure that goes from pulmonary trunk to aorta, so helps us to bypass the lungs again, but becomes less important and closes as we are born on postnatal life. If we look at the surface anatomy of the heart, we noticed that it was important to know about surface anatomy so we could learn where the coronary vessels were flowing. Word sulcus is probably familiar from your study of the brain and brain's surface anatomy. We can find sulcuses between atria and ventricles, and also between two ventricles, so left and right side. We also find grooves. And let's have a look of the most important surface anatomy landmarks. So we have something that's known as atrioventricular groove. That's the groove that separates the atria and ventricle, both on the left and right side. Sometimes also known as coronary sulcus. So that's the same term, two terms for the same structure. We also have a depression between the ventricles, both on anterior and posterior side. So this depression is known as interventricular sulcus, so between the ventricles. So that's what that means. And like I said, both on anterior and posterior side, so both at the front and back of the heart. Well, now these depressions on the surface of the heart become important when we look at the coronary arteries. So coronary arteries are what supplies the blood to the heart itself. For example, the heart muscle. And that's really the main function, important function of the coronary circulation. Heart works harder than any other organ. I'd claim I'm partial because I've spent most of my research career studying the heart and especially studying these three vessels that we'll see. So that's because heart muscle works so hard. It also will, like any other tissue, require blood flow to that tissue, both for getting oxygen there and getting nutrients there, and removing then carbon dioxide that builds up and any other waste products that build up in cellular respiration. So left coronary artery arises on the left side of the heart, right after our aortic valve. One of my favorite questions in an exam is to ask that what is the first vessel to arise after aortic valve? And that's going to be our coronary artery. Most diagrams on most textbooks don't remember to highlight that. Students often end up thinking about our other vessels that come from the aortic arch. So we're not going to be really referring to those. We're really referring more to the coronary arteries in this case. So just something to bear in mind. I'm thinking what else could I mention at this point. Well, let's just continue with our review, and if there's something else that I've overlooked, it'll come to me. So we'll notice that the left coronary artery divides almost immediately on the left side to two further arteries. First one of these that we're going to look at is abbreviated in medicine as LAD. That's called left anterior descending artery. That means that it's on the running from the left artery, runs on the front side of the heart, so anterior side, and it goes down. So that's what the name means. In anatomical textbooks, this is often rather referred as anterior interventricular artery. So it kind of makes sense that anterior interventricular artery would run in anterior interventricular sulcus. So both of these terms you might end up seeing. It depends whether you're looking more of anatomy and physiology textbook or you're looking more of a medical practice. And I've added there runs in watts. And these letters A, B, and C would refer to these coronary landmarks that we looked at, these depressions on the heart surface. Well, let's have a look at the other branch that came from the left coronary artery. This would be our left circumflex artery. So left circumflex artery keeps running in our atrioventricular groove until it goes to the posterior side. So it runs between atria and ventricle in that depression. And the third vessel that we have for coronary arteries is going to be our right coronary artery. Right coronary artery has its own branches, but at this level, we're just looking at the main three vessels. And that would run on the right side, following our atrioventricular groove again. So between atria and ventricle on the right side until branching to the back of the heart, to the posterior side. I mentioned the coronary sinus earlier on when we looked at the structures inside the right atria. So the coronary veins, they are not quite as regular like veins in general, but they are better in terms of regularity in the heart. But I normally don't spend time on the veins because in other parts of the body, they're not as reliable where they run in terms of the anatomical landmarks, and only the major veins are located where we expect to see them. So they collect the blood from the coronary circulation and dump it back to the right atria through coronary sinus. When we look at the physiology of the heart, we'll notice that heart contractions, cardiac contractions, they're of course involuntary. You don't have to think that now my heart contracts, or during your sleep, wake up for every heartbeat. That would not work. So these take place without our conscious control, but you can alter the heart rhythm with autonomic nervous system to a certain extent. We have a diagram here of the basic heart rhythm. You'll remember our action potential from the past. This doesn't quite look exactly like an action potential. It's a little bit like an action potential, but slightly tilted in terms of the shape. But let's have a look of the important structures that we see here. So, heart has a pacemaker potential, which is kind of the resting phase of the heart before we reach the critical point where the heart rhythm is triggered. And pacemaker potential was the kind of resting stage. But notice that pacemaker potential is not a flat line like in our action potential. It's slightly increasing all the time until that critical point is reached and depolarization starts. So now our electrical current shoots at the membrane to 0 and slightly above. And after reaching that point, it returns back to the beginning of the pacemaker potential charge in a process known as repolarization. So 3 steps that we want to know there, where our pacemaker potential is what we normally would see as a flat line, but in case of the heart, it's slightly increasing all the time. Quick, sharp upshoot, which is our depolarization, and quick, sharp downshoot, our repolarization. And if you were interested what happens in each of these steps, I provided further details in our lecture video. Let's have a look of then how these electrical currents flow through the heart. And this is a diagram of the heart. I think I drew it for another purpose, but I adapted for our uses here. So you'll see it looks very much like this diagram that we saw here earlier on. So we should be able to recognize similar structures. So let's have a look how the electrical currents flow through the heart. Well, electrical currents at the heart and the heart rhythm really is generated from the SA node, which is up here where my mouse is. I hope you're able to see that. So the very top part, superior part of the right atria. SA node is what makes the heartbeat. It generates this electrical control of the heart rate. And from SA node, this electrical current travels across our atria, both on the right and left side. So when the electrical current travels through the atria, this causes the atria to contract. So atria contract both on the right and left side. Then these electrical currents reach our AV node, which is the second circle that I've shown here. And this is kind of in our atrioventricular wall of the right side. This is a spot where the electrical currents are delayed, just slightly. So there's a little bit of a pause before we let these electrical currents proceed further. This pause is so that our atria have enough time to fully contract and fully empty all the blood to the ventricles. So there, it makes sense that we have a slight delay. Well, from AV node, these electrical currents travel in our interventricular septum and all the way to the apex and then up on our lateral walls of the ventricles on both sides. So there's two different parts on this part of the path. First, we have our bundle branches, sometimes also known as bundle of His. You might see that in different textbooks. We also have our Purkinje fibers. And finally, this passage of the electrical current through the ventricle wall causes ventricles to contract. So really, there's kind of a rhythm thanks to this sinoatrial node and AV node that causes the delay, that atria contract and ventricle contract. Atria contract, ventricle contract. So they're kind of rhythmed so that one is contracted, other is relaxed. And that's all thanks to this electrical conduction of the action potentials through the heart. So that's what I'm trying to show there. Some clinical considerations that relate to that. If we have irregular heart rhythm, we talk about arrhythmia. Sometimes this irregular heart rhythm can be all out of whack. So there's totally rapid, fast contractions that make no sense whatsoever. Pumping of the blood is no longer successful. Circulation is not working. So heart just goes and contracts like crazy. It doesn't help the flow of the blood since it's not synchronized. In this case, we talk about heart going into fibrillation, myocardial fibrillation. What we can do, we can use our defib, so automated external defibrillation machine. And what defib does, it delivers an electrical impulse to the heart. And what we hope to do is to stop this dysfunctional electrical currents through the heart with this electrical shock. And we hope that that gives us a clean start for our SA node to start another successful action potential series through the heart. Some patients might go or might have issues with the heart rate being maintained by the heart and with the electrical conduction, and they might have implanted device on their heart. Usually the device itself is located on the chest, and then the wires from the pacemaker wrap around the heart. So what this does, it's like a defib, but it's inside the body, so it recouples the atria and ventricle whenever there's a decoupling of those, whenever it doesn't work the way it should work. Well, I guess that the next logical thing to look there is then how do we regulate the heart rate? Well, heart rate is regulated from the brain, and if we think about it, heart rate and having successful beating of the heart is quite vital. So that's one of the structures that evolutionary were first to appear, or one of the functions that were evolutionary first to appear on the brain. And that's also during your individual development, during your fetal and embryonic development, one of the first functions that we want working in an individual. So it's located in the oldest part of the brain, within our brainstem, and especially within the medulla of the brainstem. Within the medulla, we have certain parts which are known as cardiac centers. So heart rate is regulated from there. This These signals from the cardiac centers of the medulla are passed down the spinal cord to the sinoatrial and AV nodes. Well, if you remember from our nervous system, you had a cranial nerve number 7, our vagus nerve. That was the nerve that we often called as wandering nerve. Its function is to slow down heart rate. So sometimes vagus nerve might contribute if we want to slow down the heart rate. So that might play a role in the nervous system regulation of the heart rate. Let's have a look of an individual heart cell, cardiac muscle cell, and how an action potential there looks like. Well, now again, it doesn't look like the action potential that we've seen before. But that's because heart is a little different. But we'll see that there's a resting membrane potential, which is a flat line. But once the resting membrane potential is triggered, that we are now triggering an action potential within the heart muscle cell, we get depolarization. We saw this also on the nerve cell. This is just sharper in the cardiac muscle cell. What we'll notice that before there is a repolarization, which we also saw in traditional action potential of the nerve cell, before the repolarization, so returning back to the initial resting potential can take place, we have something called a plateau. This plateau is almost flat area of the membrane potential. So membrane potential remains almost constant until we get to the point where the repolarization takes place. So the steps that we want to note there are the depolarization, plateau, and repolarization. The other thing, if you pay attention carefully to the timing, how long in milliseconds our cardiac muscle cell action potential took, and compare that to our nervous system action potential, we'll notice that the cardiac muscle cell action potential takes much longer than nerve cell action potential. We also talked about ECG or EKG, so that stands for electrocardiogram, and this is a method of measuring externally all of the electrical currents and summing them together of the heart through various different LEDs, usually about 12 LEDs. And this gives us a wiggly line that we record, and these wiggles in this line, each one have a specific purpose, what they tell us about what's happening in the heart at that time. So this very simple test, looking at the electrical currents, gives us a lot of information about the condition of the heart. We are looking whether we see these elements, and we're looking at the distances, so time delays between these elements. And by looking whether we see something, whether it's the shape that we expect it to be, and whether the spaces between these events are spaces that we expect to see, we can tell a lot about how well heart works. So we'll notice that the first bump that we see on a single trace of an electrocardiogram is our P wave. P wave appears when the depolarization of the SA nodes takes place. Also, the atria become depolarized at this point. So this depolarization causes a little bit of a bump on this electrical summation of action potentials. After that, there's a slight delay or slight resting phase, and then we quickly shoot down, then we shoot up again, and we shoot down again. Usually the second downshoot even goes a little lower than the first downshoot. But these are very closely coupled, these movements of the trace of the action potential summation. So they have been named as Q, R, and S points of this trace. And because they're so closely packed, we call this entire structure as our QRS complex. QRS complex shows us that ventricles now are depolarizing and atria repolarizing. So seeing this trace tells us about these events that are taking place within the heart. And finally, after a little delay, we have the last bump, which is slightly bigger typically and slightly longer than our P wave. This is our T wave. And T wave shows when the ventricles are repolarizing. So from those electrical events that we saw here on this diagram, we can follow them on our ECG trace here. And like I said, we are not just looking whether we see these bumps and whether the shape of these bumps is correct. We're also looking intervals between these elements. And that's how you analyze an ECG. Couple of terms that are important for us to know about the heart. You remember systole referred to the period of contraction, and diastole to the period of relaxation. And a single cardiac cycle was the one complete cycle of the heartbeats. So the blood flows now through the heart during a cardiac cycle. So that's one complete heartbeat that we're noticing in a single cardiac cycle. We also could listen to heart, again, in addition to how valves work and so on. We're looking of the two sounds. We're looking for a lup followed by dup. So it often is described the sounds that you'll end up hearing in a healthy heart as lup dup, lup dup, lup dup, lup dup. And the first sound that you hear comes from when our AV valves close. So both right and left AV valve closed approximately at the same time, and you'll hear this first sound, lup sound. After that, after a slight delay, we'll hear the second sound, which is more of a dup. And that's when our semilunar valves close. So that's our pulmonary valve and our aortic valve. So we can actually tell if there's an odd sound on the other one of these, that which valves are affected if the sound is not correct. In terms of the physiology, we also mentioned that cardiac output tells us how much blood gets pumped out within a certain period of time. In this case, we're looking within a minute. So cardiac output is calculated by multiplying heart rate by stroke volume. Heart rate is typically about 75 beats per minute. Stroke volume is how much blood was pumped out in a single contraction, and that is typically about 70 milliliters. So if you do the math, we end up to roughly about 5.25 liters per minute as a typical cardiac output at the resting individual. If you start exercising, the cardiac output will grow as your heart rate goes up, and your stroke volume can go up as well. And we can get to about 20-25 liters per minute. Athletes can get to 30 liters per minute as well. So it depends a little bit about your fitness level. But that gives you an idea that our heart really allows us to adjust to the need of the body. I think that that's quite a significant increase in my mind. I'm pretty impressed about that. Clinical relevance, the last thing that I mentioned here, we talked a little bit on our lecture material about congestive heart failure, CHF. You might want to have a look of it, but really that's something that I think that we've covered fairly well. Really the issue here is when the heart does not work properly, we constantly have elevated blood pressure. A lot of the muscle might have been scarred, so it's not functioning as well. So we end up not pumping enough blood out of the heart. So the blood ends up building before the heart, and it can end up filling lungs or causing an edema somewhere else in the body. Imagine the way to die, that your lungs get filled with fluid, your own blood, or elements from your own blood. That would not be a great way to go. So your weak heart is not working quite as well as it should. So the blood doesn't get pumped out properly. How do you treat congestive heart failure? You want to drain some of the fluids out and use drugs to increase the contraction of the heart. I didn't get a chance to speak with you about embryology of the heart. That's somewhat one of my favorite topics, as I spent a lot of time during my honors years looking at the embryology, especially comparative embryology, and how heart develops. Heart is one of the first organs, and there's a lot of great information available on your textbook if you're interested in that, but that's not something that I would really expect you to be super experts of. You'll notice that I'm mentioning here again, though, that bypassing of the lungs during embryonic development, because the lungs don't really work. Why would we pump that much blood there? That was our heart chapter. And the last chapter that we have to look, where you don't have answers in your review sheet, is gonna be our blood vessels. If you have any questions about the heart, this is a good time to shout those out, but of course you can shout out them later. You can also ask questions at the end, but I think that if I don't hear anything, I will move on to talk about the blood vessels, since we started late and I don't want to keep you too much over time. I'm just gonna jump ahead, but if you need anything more, give me a shout out. I'm happy to answer always questions. So in terms of the blood vessels, we noticed that there were 5 different kinds that we need to know. Blood ran from your heart first to the arteries. These were the first vessels leaving the heart. Arteries then turned to arterioles, which were basically smaller versions of the arteries, and then we get to the capillaries. These are the main side of gas exchange. Other thing to note with the capillaries is that we have to have a very low blood pressure at the capillaries because they are weak, because they're so thin. If there's high blood pressure, that ends up breaking the walls of the capillaries, and we don't want to do that. That would not be good. So the blood pressure is high as blood leaves the heart, but by the time we get to the capillaries, blood pressure has dropped significantly. And then from the capillaries, blood returns back to the heart through venules, which were basically just at our discussion, smaller versions of the veins, and then veins, and eventually to the heart. The order of these vessels is important because that shows that you know the flow of the blood as well properly. So be familiar with those. What else we noticed? We noticed that all blood vessels had 3 general layers. There is an exception of our capillaries where we don't see all of these 3 layers. But in all other blood vessels, we do end up seeing these 3 general layers. The thickness of these layers may vary. And we're going to look through these general layers starting from inside the blood vessel. So from the lumen of the blood vessel. So the innermost layer, the first layer that we'll see if we go out from inside the blood vessel, is going to be our tunica intima. And intima, how I think of it, is that the blood is in intimate contact with this layer. So what we see here, tunica intima is made of simple squamous endothelium. So these were the one layer of flattened disc-like cells. I think that at the end of my lecture video for this topic, if you did not remember what these tissue types that we had for epithelium, I added a link there on the end of the video, so you can have a look of revision from one of my Bio 156 or Bio 201 classes, or even Bio 181 class. I cover it there. Tunica intima, really its function is to protect the other layers, and it is very thin, single layer of these cells so that the materials are able to pass through this layer easily. Well, the middle layer of the blood vessels is called tunica media. Tunica media is the thickest layer, the middle layer, and there can be either smooth muscle or elastin, or both. And the proportions of these change depending on the kind of a vessel that we're looking. The functions of that is the tunica media is able to contract and dilate, so change in terms of its diameter as needed. And we remember vasoconstriction referred to when the diameter of the blood vessel decreased, and vasodilation when the diameter of the blood vessel increases, grows. So let's have a look of the last layer, tunica externa. Externa, I think, is a word that gives us a clue that it's the outermost layer. Often, tunica externa can be also described as tunica adventitia. depending on where you study, but both words would be useful to know. The function of the outermost layer is really to support rest of the blood vessel, reinforce the walls, and anchor these vessels to the site where they're at. This is made of largely of connective tissue. We end up finding also nerves and lymphatic vessels running within the tunica externa. Also, another thing that we end up finding on the outermost layer, the tunica externa, is blood vessels itself in the blood vessel wall. These are, especially in very large vessels, we end up finding these little blood vessels. And the function of these little blood vessels is to supply the blood vessel wall itself. These are known as our vasa vasorum. Their function is to nourish or supply the blood vessel wall itself. You can consider them a little bit of an adaptation similar to the coronary arteries. So we're making sure, since now we're so far from the inside of the blood vessel, this external layer is not able to get blood from the inside the blood vessel necessarily as easily as tunica intima and tunica media. So we facilitate a flow of blood to the tunica externa by having little blood vessels supplying that. And remember, those were our vasa vasorum. Then we went through the different blood vessel types, and I'm going to go through this fairly quickly. With the arteries, we remember the arteries were the vessels that arose immediately after the heart. The most close artery to the heart would be, of course, our aorta. So these are exposed to high pressure because you remember the left ventricle contracted really hard. So now the blood is there pushing really hard towards the walls of the blood vessel. So arteries are in general very thick walled and they're very elastic walled. So they're able to handle that fluctuation of the blood pressure, but also they remain in their shape even if we drain the blood out of the vessels. And that's if you end up doing, for example, postmortems or so on, looking at the dead animal where the blood has been drained out. You'll notice that the arteries are always much more regularly shaped, better shaped. The function of this elastic thick wall is also to even out the pulsatile flow of the blood, because we remember the left ventricle squashed the blood out and then relaxed, squashed the blood out, then relaxed. So throughout the arterial network, this flow now evens out once we get to the very end of the arteries. And there's actually only three if we if we consider also our arterioles, but let's talk about arteries as two different types of arteries that we need to know. So the first types of arteries that we need to know are the arteries that are closest to the heart. These are known as elastic arteries. Their function is really to conduct the blood to other parts of the body. They have a thick wall. Their lumen gives us very, very low resistance for the blood flow. So the flow of the blood is really easy inside these elastic arteries. They contain a lot of elastin, which is a clue about their name. And really, we don't end up finding vasoconstriction happening within the elastic arteries. They do expand and recoil as the blood is at this point very pulsatile. And in that sense, they kind of become pressure reservoirs. So over time, because they do that, the pressure becomes more even in other parts of the arterial network. Once we move further downstream on the arteries, we end up to muscular arteries. These play a role more of in distributing the blood to the other parts of the body. So they're no longer just conducting, but now we are starting to do distribution to many different areas, many different parts of the vascular network, and they are often referred as distributing arteries. Most of the arteries are muscular arteries. What we end up finding in their structure is that the tunica media, which we just had a look of, that's very thick. There's a lot of smooth muscle and less elastic muscle. So opposite to what we saw just before. in the structure. And their function is now to do the vasoconstriction, which we didn't see in the elastic arteries. And the last type of arteries that I've listed here, which I think I could have listed as a separate group, is our arterioles. You remember these were just smaller versions of arteries. So single layer of thickness in terms of the number of cells that we end up finding there. And really the big thing to note about arterioles was the 3 capillary sphincters that we found on these. So right before we go to the capillaries, we have these little bands of smooth muscle around blood vessels, around our arterioles, that either can be squashed or open, and they control where the blood flows. And we can either open up a set of capillaries or we can close them off. So we direct the blood wherever it's needed. So that's one adaptation of the arteries that we want to know. The most important thing about capillaries that I want to highlight, blood pressure there is very, very low. That's because the wall of the capillaries is very thin, so we cannot handle high blood pressure there. They're small, very microscopic vessels. The smallest of the capillaries have a diameter of approximately 1 red blood cell, or even a little smaller, because the red blood cell can actually get squashed a little and it will still pass through the vessel. Within the capillaries, we only see the tunica intima, so the innermost layer, and the function of the capillaries was really the exchange of the materials. This was the main site of exchange of the materials. That can be gases, that can be nutrients, that can be waste products, that can be hormones, that can be a lot of things that are being exchanged at the capillaries. And there were 3 different kinds of capillaries that I wanted us to know. And if you look at these 3 different capillaries as a whole, you'll notice that this listing goes from capillaries that have almost complete wall to the capillaries that have huge gaps or huge parts of the wall missing. And the amount of wall tells how much and how easily stuff gets exchanged from capillary to outside and from outside to the capillary. So continuous capillaries have the most complete wall, as you can see from this picture or diagram that I included there. We end up finding continuous capillaries, especially at the skin, muscles, lungs, central nervous system. There is a special kind of barrier. So we want to keep our brain and blood that flows within the brain separate from the rest of the circulatory system. So we end up finding blood-brain barrier at that site. So this doesn't facilitate this blood-brain barrier direct passage of materials, but really what I want you to know is just that we have a continuous wall layer of the tunica intima in these continuous capillaries. The next type looks a little bit like Swiss cheese. This is known as fenestrated capillaries. And remember, the word fenestration meant a window. So fenestrated capillaries have small openings in the capillary wall. For example, you find fenestrated capillaries at the kidneys, intestines, where hormones are secreted. So it allows more materials to pass through this kind of capillaries than through the continuous capillary walls. But the last one, it looks like there's huge parts of the wall missing. So really big sinuses. And you remember sinus was an opening of some kind. So sinusoidal capillaries have big, big openings there. Because they're so big openings, this also slows down the flow of blood through the capillaries. So the resistance grows up since it's no longer nice and smooth layer, as you could see in the continuous capillaries. There was a nice and smooth layer, so flow is quicker there. But really in the sinusoidal capillaries, the flow slows down significantly. And this is ideal for these parts because slower the blood flows, easier it is for us to do these exchanges of materials in these types of capillaries. We find sinusoidal capillaries in liver, bone marrow, spleen, adrenal medulla, places where we want to do especially lots of exchange of materials. I mentioned here again the precapillary sphincters. We had a look of those before, but this shows that this is a really important concept. So precapillary sphincters, opened or closed networks of capillaries. So if we have a network of capillaries that are all interconnected, that's our capillary bed. That's the term that we use to describe those. Oh, I didn't mean to zoom in. Sorry about that. And then let's have a look of our veins. Our veins have lowish blood pressure. It actually picks up the blood pressure a little bit from what we had at the capillaries, but it still remains fairly low. So you remember the big thing is that we need to drop the blood pressure before we get the capillaries so we don't break their wall. And actually, majority of your blood vessels are going to be veins. And the big concept that I discussed with you guys in terms of the veins was a concept of venous return. So this is all about how the blood flows back to the heart and sometimes, for example, even against the gravity. So this is because of two structures, valves that we find within the veins and also skeletal muscles. So skeletal muscles end up squeezing our veins. And remember, veins had thin walls. And while the veins get squeezed, some of the blood gets pushed to the direction where we want the blood to go, and it doesn't get to come back because we have valves that control that the flow is going to be one directional, unidirectional. So when your valves failed in venous return, you ended up with varicose veins. This was also where we discussed about the example of a soldier who stood for a long period of time, for example, in a parade with the knees locked. If we are actually normally constantly moving a little bit, and this allows us to have a venous return working constantly. And if we end up with this movement being prevented, we end up then with insufficient venous return. Enough blood isn't returning to the heart, and eventually your brain will start to slowly get depleted of the sufficient blood supply, and your brain will react to that immediately and you will pass out. Passing out was useful because now we're evening out, the body is flat and blood can flow really easily to the heart and to the brain. So it's almost like a protective mechanism. This would make a really good extra credit question to discuss venous return, what it is, and how it related to this soldier that passed out. If my explanation here didn't make sense, I've discussed it quite nicely in one of our lecture videos. Have a look through on the module where we talked about blood vessels. But that would make a good extra get credit question. Few terms that relate also to our veins. Veins have a lot of anastomosis. Anastomosis were these connections with the blood vessels for lateral circulation. The function of anastomosis is that we can find an alternative route for the blood to go from our arterioles to veins or venules. If one part of the blood vessel is blocked, for example, here in the arteriole, we have an alternative way so the circulation still remains. We just skip that part that's blocked. In terms of the physiology of the blood vessels, a big thing there that we discussed was blood pressure. Blood pressure tells us how much force we're exerting against the vessel wall, and typical blood pressure is somewhere around 120/80. So we get two values. The first value is the blood pressure during systolic period, so during our contraction, and second value is during diastole, so during the relaxation. And blood pressure was measured as millimeters of mercury. There's food factors that affect the blood pressure, cardiac output, peripheral resistance, and some patients, some individuals have a condition called white coat syndrome. That's when there's a change in blood pressure when you are at the doctor's or you see a medical doctor or other health professional, all of a sudden your blood pressure is higher than normally. That happens to quite a few people. So really to get the best idea of someone's normal blood pressure is to have them to measure it themselves while they're relaxed at the home. The other concept that we talked about was pulse. Pulse tells us the number of times that the heart beats per minute, so that gives us our heart rate. These terms can be used interchangeably. And as the ventricles push the blood out to the aorta, to the systemic circulation, that's when the blood pressure shot up. And when ventricles relaxed, there's no longer blood being pushed out to the systemic circulation, so blood pressure drops. It's this variation between these two that we can feel as the pulse, because pulse is always measured from our arteries. There's two different places where you can measure pulse: from your neck as carotid pulse, from your wrist either as ulnar or radial pulse. So there's ulnar and radial arteries running at the wrist. Your brachial pulse from your cubital fossa, so from the opposite side of the elbow. From your legs as a popliteal pulse, from the feet either as posterior tibial pulse or dorsal pedis pulse, in your groin area as femoral pulse, on your temples, so temporal area of your skull, either a superior temporal pulse or external maxillary pulse on the maxillary bone. So those are some good places. I think quite common ones are either ulnar or radial or brachial pulse. Also, the carotid pulse is pretty common one to take. Short-term regulation of the blood pressure. We had this diagram that combined the homeostatic mechanism with then adding the information as it relates to the blood pressure regulation. So that I discussed quite extensively in our lecture video. Long-term regulation happens through the kidneys. So remember, kidneys' main function was not to make pee, but it was to control blood pressure. And this can be done by controlling the amount of fluids in your body, so the volume of the blood in your body, and that through that we are able to change the blood pressure. And there's two mechanisms that we might use: direct and indirect mechanism. I didn't expect you to know much about them, just that they exist. The indirect is also known as renin, angiotensin, aldosterone mechanism. So really, to summarize, we want to have high enough blood pressure so that the tissues actually get perfused, but we don't want the blood pressure to be too high so we don't end up breaking the blood vessel walls. And I think that I discussed about hypertension and hypotension. So blood pressure is affected by many factors. If we have high blood pressure, we talk about hypertension. And this can have many problematic consequences: heart failure, vascular diseases, renal failures, even stroke. And many factors contribute that, but often we can't pinpoint a single factor, but it's actually a combination of many factors. And there are all sorts of treatments that we can do. Medication is not the only one by any means. Lifestyle changes are a great way to treat hypertension. Hypotension referred to low blood pressure, and there's different reasons that can cause hypotension. You might have a temporarily low blood pressure. This is when you get kind of, for example, dizzy when you're standing up. If it's temporary, it usually causes less concern to us. Chronically low blood pressure, often related to poor nutrition, and that has risk of other conditions. And if you have acutely low blood pressure, you might be going into the shock. But in general, consistent hypotension or temporary hypotension is not really as risky as hypertension. Some research has even proposed that it might be even beneficial to have slight hypotension. I talked about the shock. The mnemonic for that is PC, fat, so police constable, that's fat, very British abbreviation. So signs and symptoms of shock: the patient is pale, they might be cold, their skin might be clammy as well, their breathing might be fast and shallow, or the pulse fast and weak, they might appear agitated, thirsty, and that's a sign that they're going into a shock. And I listed there a little bit of first aid as well that you can do. So those are signs and symptoms of the shock. I'm all about in our classes that we should know a little bit of first aid, not just what happens, but we can do something at our level. First aid saves lives. There's extrinsic and intrinsic control of the blood flow. We really didn't talk too much about that, but then we had a look of special circulations. So notice that there's local factors that play a role in circulation a great amount. It's not just the entire global circulation that's changed, but we changed locally circulation based on the needs. This is known as autoregulation. And then we went through different special parts where circulation needs to be different. I do encourage you to have a look through these diagrams that I provided. They are the same that you had in the vasculature chapter, but they might be handy because especially on your anatomy lab practical, you do end up having some blood vessels to know. So these summarize them nicely here as a diagram, very schematic view. Or, so I have both arteries and veins, major arteries and veins of the body. And then I have a nice artery diagram that's more of a laying it over the outline of the body. And then I have different special circulations. So for example, circulation to the brain and head in general, both as a schematic diagram here and as more of an anatomically accurate diagram. Circulation of the brain also here, looking from the inferior side, looking from down towards the top. Here I have upper limb schematic diagram of that again, followed by more anatomically correct diagram. I have similar for the abdominal region, and again, more anatomical diagram on a few different levels for the abdomen. Same thing for the lower limb schematic, followed by anatomically correct view. I don't expect you to really memorize each one of these, but I think it's good that you have a general appreciation or you're able to find these as needed. If you are going to a field such as vascular sonography, then you will end up having to learn this. But at this level, really what's important for me is that you have a general understanding, general appreciation, and then you can study specific blood vessels later. What is important to know in terms of the blood flow is that the speed of the blood in the cardiovascular system, in the different parts, either in our arteries or in our veins or in our capillaries, is dependent on the cross-sectional area of that region. So as the cross-sectional area, total cross-sectional area increases, the speed of the blood decreases. So the slowest speed is at the capillaries because that's the total area of the vascular bed in the capillaries is the largest. And that makes sense because we want the blood to flow slowly through capillaries because that's where the exchange happens. You remember there were two forces working on the blood vessels, our hydrostatic and osmotic pressure, and we had a look of those also more when we started to talk about lymphatic system. Clinical relevance is edema if you have buildup of fluid when the hydrostatic and osmotic pressures are not balanced. So when we end up with extra fluid outside the blood vessels. Key points really to take away is that arteries run deep, veins are more superficial. We have alternative routes on the veins. These were known as anastomoses. And then I have all the answers on your review sheet for the lymphatic and immune system because we started so late today. So this should help you to revise those. Tonight, I probably am not going to go through these unless there's a great interest in that. These are something that's fresh in our mind. I do want to say in terms of the lymphatic and immune system that we covered them only on very general levels, so I don't expect you to be amazing with these, but really to kind of just have a basic understanding. In terms of the lymphatic system, I'm just going to do really quick run through. You notice the lymphatic system is linked also with circulatory and immune system. The terms plasma, interstitial fluid, and lymph, and where to use each one of these terms, that's important. That's something that I like asking because it's such a fundamental concept to understand. So plasma is part of the blood, And when some of the blood leaks out of the blood vessels, it's usually from the plasma, or it is from the plasma, and it becomes interstitial fluid. So the composition of the interstitial fluid is exactly the same as plasma. When interstitial fluid gets picked up by the lymphatic system, we start talking about lymph. And again, composition of lymph is the exact same as interstitial fluid or plasma. So it's all the same stuff, just called different depending on where we are at. We compared lymphatic and cardiovascular system. We noticed both of them are one-way systems, but whereas cardiovascular system was a closed loop, there's no clear anatomical openings. Lymphatic system was open from one end and then dumped the contents of the lymphatic system to close to the heart. So it's open where the capillaries are. So we're collecting the lymph from there, any tissue or any interstitial space. The functions of the lymphatic system, 3 important functions: fighting against diseases or pathogens. So our white blood cells within the lymph nodes do this job. Also transporting fluids. So this is the extra fluid that leaked out from the cardiovascular system. The last one was something that often gets overlooked, and it is not the most important topic, but it's a topic that will run again when we talk about digestive system. And I like asking about this because I think that this is just because we often ignore it. We need to start highlighting it more. Because blood vessels are limited on the size of molecules that they can pick up. For example, from the food diet that goes through the intestines, goes to the digestive system. But molecules that are too big for the cardiovascular system to be picked up can be picked up by our lymphatic system and then transported within the lymphatic system, within the lymph, until we get to the very close to the heart where we dump the contents of the lymphatic vessels at the level of subclavian. This can be good that we pick up large molecules. For example, we can pick up large proteins, cell fragments, pathogens that we want to pick up and get into the lymphatic system so we can deal with those. But it can be also bad if pathogen gets into the lymphatic system and spreads there uncontrolled. Or, for example, if you cut into a cancerous tissue and some of the cancerous tissue ends up within your lymphatic system, there is a risk then that those cancer cells might end up spreading throughout the entire body. And for that reason, a lot of the time, some patients end up choosing that they don't want surgical intervention for their cancerous growth. In terms of the lymphatic vessels, we start with the smaller and go to the bigger. So you'll notice lymphatic capillaries, they were open from one end and they're much more permeable than blood vessel capillaries. They're made of simple squamous epithelia, so single layer of these flattened discs, even easier than in the blood vessel capillaries for material to pass across those. Then lymphatic vessels grow in size. We end up with collecting vessels. They are thin-walled in general, lymphatic vessels. They have valves, just like our veins had, and they have anastomosis, so a lot of those interconnections. I mentioned the posterior thoracic cavity, how thoracic duct ran through it, and at the bottom, so inferiormost part of the thoracic duct, we had cisternaculi, the dilated sac there. Then we end up with the trunks, which are again bigger in their diameter. We had lumbar, interstitial, intercostal, bronchiomediastinal, subclavian, and jugular. So where each one is located, I've also listed there. So lumbar at the lower back, intestinal, of course, intestines, intercostal, between the ribs, bronchomediastinal at the region of bronchiomediastinum, subclavian below the clavicle, jugular at the neck or throat region. And finally, the biggest ones are our ducts. So we end up, for example, with the right lymphatic duct that drains the right arm, the right head, right neck, and empties to the right subclavian. Thoracic duct was another one that I mentioned there as an example. I mentioned a few times that the composition of the plasma and interstitial fluid, and lymph was the same. So if you remember from our discussion of the plasma, it's the exact same. We have within the lymph water, proteins, white blood cells, and then a few other things such as salts, glucose, and fats. But those three are the most important. We should never find red blood cells in the lymph. If we do, something's wrong. Lymph is produced initially at the bone marrow and lymph nodes. And the clinical relevance that I wanted to mention was lymphedema. So again, building up a liquid, in this case, lymph, that caused swelling. You remember it was all usually one-sided, not on both sides of the body. It wasn't symmetrical. Can be due to tumors or removal of tumors or removal of lymphatic vessels. But usually body is pretty good at adapting this if we end up having other areas or other lymphatic vessels picking up their role. Lymphatic cells go, even though they started at the bone marrow, they go to mature if they become immune system cells proper as lymphocytes either to the thymus, and in this case they'd be called T cells, if to the bone marrow as B cells. Then we also had supporting cells within the lymphatic cells. Lymph nodes were important. These were small, less than 1 inch in diameter glands, kind of a bean-shaped, located as chains within the lymphatic vessels. There's hundreds of these in the body. Their function is to filter lymph, but also house the immune system cells, so white blood cells there. And these immune system cells, of course, fought against pathogens. When there is an inflammation, lymph nodes tend to swell, and that's what the doctor was doing, checking those. So inflammation is easy to pick up from the swelling of the lymph nodes, and then we know that somewhere in your body you have inflammation going on. Where you might find groups of our lymph nodes, good areas are, for example, at the neck, the cervical lymph nodes, at the armpit, axillary ones, supratrochlear lymph nodes, above the trochlear notch here, and your groin areas, inguinal lymph nodes, abdominal area, abdominal lymph nodes, thorax, thoracic lymph nodes. You remember single lymph node in terms of its histology had both cortex and medulla. Within the medulla, we had sinuses. So those openings, any opening in the body is known as sinus in anatomy. So in this case, these are openings within the lymph node where the macrophages live and they screen the lymph flowing through. Here's a diagrammatic representation of a lymph node. Notice that blood is coming in this diagram from our right side to the left side. So the vessels that bring lymph to the lymph node, they are afferent, and vessels that carry the lymph out from the lymphatic node, they're efferent. And in between, there are some connections there as well. And we talked about the smaller lymph nodes as a sign of infection. The lymph nodes end up growing in size because the amount of white blood cells in them grow. So that's why they are large, but they also feel sore because we're now stretching this tissue. So there's more white blood cells than normally. So it feels a little uncomfortable. I think we finished this part by looking at 2 different lymphatic organs that were most important. So spleen, that's the largest lymphatic organ, about fist-sized, located only on the left side of the abdominal cavity, just below the stomach. Remember, patient's left side. There's a very rich blood supply to the spleen, so you notice there's a splenic artery and vein there. The function of the spleen, immune surveillance, immune response, cleaning blood, so we remove old or dead blood cells, platelets, and such. At the spleen, also storing some of these components such as iron and platelets so we can reuse them. And in embryo, we had red blood cell production initially at the spleen. The other lymphatic organs that we discussed were our tonsils. So they are named of the location. We have palatine, lingual, pharyngeal, and tubal tonsils. If you have inflammation of tonsils, that's tonsillitis, and some of us have had tonsillectomy, removal of the tonsils surgically. Appendix, that was the little expansion on the first part of the large intestine, so hanging out from the cecum. It can get inflamed, and then the appendix can be removed. Really, the function of appendix, we don't think that it has a function these days. We are not certain. The best explanation at the moment is, though, that it would be evolutionary remain. That's where the bacteria that was used to digest plant-based material was housed back in the day. And now that we don't eat as much plant-based material, it has started to shrink, and eventually probably disappears. Thymus was the last one that we mentioned about lymphatic organs, large in infants and during puberty, but then it disappears or gets smaller, replaced by connective tissue and adipose tissue, until on an old person it's hard to find thymus. Thymus is still functional, just less active, but it still functions. Immune system, I really have very, very little notes about that, because all I want you to know is that it is more of a functional system. It is really the white blood cells within our lymphatic system form our immune system. And its main function was to defend us against pathogens. So any virus, bacteria, parasite, and so on that might be in the body, invading body. But what pathogens want to do, they just don't want to get into the body, but they also want to multiply so they can take over. So if you think of that cartoon, what they want to do, they want to take over the world. And how they are going to do that, they're going to multiply. There's two branches of immune system: innate immunity, which we looked first, that was very non-specific, general response, protecting against many different kinds of pathogens. And we had 3 lines of defense there. We had our first line of defense, which was having physical barriers, so preventing the pathogen from ever entering the body. So for example, skin and mucous membrane were good examples of that. Second line of defense, various different chemicals, so sweat, stomach acid, tears, urinating, and so on. Those can also keep the pathogens away if we are unable to provide the physical barrier. And then we have cell responses, and we discussed of all of these responses in a little bit of detail. One important one that we discussed at this point was the inflammation, signs and symptoms of inflammation. I would know, so if an area is inflamed, it's usually red, it's warm locally, it's swollen locally, and it might be painful as well. I gave you a little bit of explanation why you should use word purulent rather than pus in our lecture video. And then I do want you to know that what fever is. Well, fever can be defined as an abnormally high body temperature. And then the second branch of immune system was our adaptive immunity, often in Europe called rather as acquired immunity, which I think makes more sense. So that's all about these memory cells. If we have faced a pathogen before, now we have generated a memory against that. So if we face it again, we have a stronger attack against it. However, this took time, so this adaptive immunity is poor on the first encounter with a pathogen. It targeted very certain antigens, so you cannot have adaptive immunity against every possible antigen until you've met every possible antigen. And in addition, that doesn't act only locally, it acts throughout the body. We talked a little bit about humoral and cell-mediated branches of the adaptive immunity. We talked about B and T cells, we mentioned them already earlier on. And we also talked about the natural and artificial acquisition of the adaptive immunity. In terms of the clinical aspects, we talked about immunodeficiencies, Hodgkin's disease, AIDS, for examples that we looked, many autoimmune diseases, we had a look of them, and few hypersensitivities as well. So that's shows the kind of relative weights of these chapters. So immune system is really just basics that I want you to know, whereas then the lymphatic system, to a certain extent, I want you to know it a little bit more than the immune system, and then I want you to know the heart and blood vessels really well. So that was our review sheet. And the big topic that we cover in these review sessions. Like I said, I will be removing the answers or the version with the answers for the last chapter later on. So if you want to download it for yourself, since you were here on this early session, I really am quite happy for that. And the last thing that I want to do is just share quickly from our PowerPoint the last bits of this session. So let's get that to a full screen. So there is going to be a lab practical review sheet as well. I have at the moment it in 3 parts. I might look of doing some sort of a condensed or combined version of that. Remember, you can practice for lab practical. Remember, it's going to be typing in answers. I will not penalize for spelling errors. If you don't know an answer, give me something, because I will always give points if there's something that I can give points for. And you can tell me about something that relates, or even what it sounded like the term, or tell a structure that's close to it. Tell me something so I can be like, you might not get full points, but you get some points. So I always encourage you to answer to those. You have about a minute for each question. That's ample time. Like I said, there's going to be lecture slides, handouts, PDFs of those. There is there is tutoring available. I'm doing these revision sessions also on Wednesday, both in the morning and evening. So at 10 o'clock and at 8 o'clock in the evening. There is tutoring available. I can do individual tutoring if you contact me. And also we have the Cranium Cafe, which is offered by the college. There are our full-time tutors or part-time tutors that you normally have also available. There's science tutoring as well, that you can go and just log into that link for the Cranium Cafe. Really, that was all I was planning to tell. If there's any questions, I'm super, super happy to answer those. If not, that's all I have for today. And I'm going to stop the recording now. So in case there's any questions, you can ask also them outside the recording. But for now, this is all I had for today.